Hello, my name is Michelle Coleman. I'm a junior at West Virginia University in the exercise physiology and psychology majors. If you think like me, you might have asked yourself before, why do people behave or think differently? My research involves looking at how autism spectrum disorders, or ASD, presents itself using resting state functional MRI technology. ASD is a very common neurodevelopmental disorder that affects 1 in 54 kids by impairing social and sensory processing. To better understand people with ASD and get earlier behavioral interventions, we need to understand what's happening in their brain to perhaps find diagnostic markers. By the end of this, you should hopefully understand more into the differences between an autistic and typically developing, or TD, individual. Using different neuroimaging techniques such as functional magnetic resonance imaging, or fMRI, we can see how a behavior or thought process presents itself in the brain. Active parts of the brain are shown on the scan through blood oxygenation differences with a task. However, we specifically looked at resting state fMRI, which looks at what regions of the brain light up at rest without a task. By comparing the resting state of someone with ASD and someone without ASD, we can better understand a baseline of what someone who develops typically brain looks like and compare that to someone with autism, and then understand how that change could manifest in certain behaviors or social impairments. Deliberate regions of interest, or ROIs, were chosen to look specifically at what those with ASD have the most difficulty with and were previously published in autism research. Autistic individuals struggle with social interactions and oftentimes present with repetitive behaviors. So regions we chose had to do with social processing, like the posterior insulae, regions involved in motoric behaviors in the basal ganglia, like the putamen, the sensory switchboard known as the thalamus, as well as body area processing, like the extrastriate and fusiform body areas. These are shown in the brain in figures 2A through C. Our hypothesis of the motor-related region interacting differently with the social processing region, this corticobasal loop, in the ASD group was not supported. However, our analysis found there to be differences in information flow between groups, and these were replicated in another set of participants matched to our WVU participants. The novel analysis used looks at positive and negative path effects in the brain. This finding is suggestive of the flow of information in the brain between regions through excitatory and inhibitory effects. Looking at figure 3A, you can just at a glance see the difference between the left ASD and the right TD panel. The ASD group had less path effect connections, the arrows shown here, than the TD group, indicating weaker correlations. Interestingly as well, the TD group had predominantly negative path effects, whereas the ASD group had mostly positive effects. Why this is occurring is still undetermined, but could relate to inhibition that the ASD group lacks. Without the same regions communicating with each other, autistic individuals could be physically unable to function the same as someone whose brain developed typically. We then proceeded to try and validate our findings using the Autism Brain Imaging Data Exchange, ABIDE. This database has thousands of participants, both with and without ASD, from all over the country's resting fMRI images. This data exchange allowed us to age, gender, and IQ match our participants to those in the exchange and see if this finding and the analysis can be replicated. This can be seen by comparing figure 3A with figure 3B, specifically looking at the left sides to compare WVU's ASD group to ABIDE's ASD group. One red arrow indicating a negative path effect can be seen for both of the ASD groups from the left FBA to the left EBA. These are two body area processing regions, indicating this perhaps is a key connection. To compare the TD groups, WVU versus ABIDE, look at the difference between the right sides of figure 3A and B. At statistical significance, both sets showed stronger correlations, seen visually through more arrows, for the TD group as compared to the ASD group. More negative path effects again were shown in the ABIDE group for TD population. The results are not identical between datasets, but these similar trends in important regions for ASD determination help us believe in this analysis and result. Looking at how this analysis can be validated further will be a next step. Other networks and regions can be looked at to develop a whole brain finding for diagnostic markers. ABIDE can be also used to address possible age and gender differences that our participants could not, because we had a limited data set of only 5 females in each group and mostly young adults. Hopefully, by validating these findings, we'll be one step closer to determining physiological differences in autistic individuals. Thank you.